In this presentation, we are going to look into structure and function of myoglobin and hemoglobin. So first you need to know how to draw correctly a porphyrin ring, which is the active part of myoglobin or hemoglobin molecule. That's what oxygen bonds to, or actually iron that is attached to porphyrin ring serves as bonding site for oxygen. So to draw a generic porphyrin ring, just minimal porphyrin ring, takes 6 easy steps. This is actually not something I came up with. Somebody else figured this out, and I don't remember who it is, so I can't give that person credit. But again, I can say that it's not my idea. So we start by drawing 4 nitrogen atoms in a square. So very simple first step. And then we draw a 5-membered ring, or make that nitrogen atom a part of a 5-membered ring. And you can see how those 5-membered rings should be oriented. Next, in the third step, we connect each of those 5-membered rings to the neighboring 5-membered ring with a single carbon atom. Of course, these are line formulas, so you can see how that connection works. These 3 steps were really easy ones. Next step is first one that is a little bit tricky, and that's placement of the double bonds. So note how double bonds are placed. They are not symmetrical. So it's easiest to first introduce double bonds on the side chains, not on the rings, not on the 5-membered rings, but those what we call exocyclic double bonds, exocyclic to the 5-membered rings. So one of the 5-membered rings, in this case it could be anyone, but in this case it's 5-membered ring in the upper right corner, will have 2 double bonds attached to it. So 2 exocyclic double bonds. 2 double bonds are attached to that 5-membered ring, which means that both of the bridges that connect that 5-membered ring to the neighboring rings have double bonds. And then 5-membered ring directly opposite to it, so that one in this case in the lower left corner has no double bonds attached to it. The remaining 2 rings, one in the upper left and lower right corner, have 1 double bond attached to each. And you can see how they work. They point towards, each of those double bonds points towards the ring with no double bonds. And anyway, you couldn't introduce these 2 double bonds in the wrong way, because then they would bump into existing first 2 double bonds that you drew. So that was the difficult part, introduction or drawing of 4 double bonds that are part of bridge chains. And then double bonds that are parts of the ring. So each ring has its own double bonds. So ring in upper right corner that has 2 exocyclic double bonds has only 1 double bond. And you can see its double bond that is conjugated with existing 2 double bonds. A ring directly opposite to it, one to the lower left corner, is typical pyrrole ring, if you know what pyrrole is, and it has its own 2 double bonds, the way pyrrole ring would have. And then remaining 2 rings have double bonds the same way in the ring, connecting 2 carbon atoms the same way as the first ring. So connecting carbon atoms 3 and 4 of the ring, or directly opposite to nitrogen atom, and in addition, nitrogen atom of those rings also forms a double bond. So when you place double bonds in the ring, 3 of the 5-membered rings have 2 double bonds each. And 2 of them, one in the upper left and lower right corner, have double bonds that are carbon-nitrogen double bonds, while one in the lower left corner has only carbon-carbon double bonds. And then the final step is placement of hydrogen atoms. You will note that 2 nitrogen atoms form only 2 bonds, have only valence of 2. Nitrogen atom has valence of 3. So those 2 nitrogen atoms will be bonded to hydrogen. So that's where we place 2 hydrogens. Remaining 2 nitrogen atoms have valence of 3 satisfied, because each one of those 2 nitrogen atoms forms a single and double bond, so their valence is satisfied, so there are no additional hydrogens. And so this is it. Now you have properly drawn porphyrin ring. Of course, in proteins, where porphyrin ring is prosthetic group, there will be additional various substituents, and you will see some examples very soon when we cover molecule of myoglobin. Myoglobin is a relatively simple protein. It's composed of 153 amino acid residues, and it's single chain. That's what makes it relatively simple. It contains heme as prosthetic group, and that's its active site. Heme binds to oxygen and allows myoglobin to perform its biological role. Myoglobin is present in the cells, and it serves as oxygen storage in the cells. It's typically present in muscle cells, but one can find it in other cells. Hemoglobin does have relatively high affinity for oxygen, and actually heme molecule itself, has relatively low affinity for oxygen. And the reason for it is that heme is easily oxidized to iron 3. Iron molecule in heme is iron 2. It has to be iron 2 plus so that it can bind to oxygen. But under atmospheric conditions, iron 2 is easily oxidized to iron 3, and it loses iron 3. With iron 3, heme loses its ability to carry oxygen. When heme is surrounded by protein chains, by protein alpha helices, it's stabilized and oxidation is prevented. And then such molecule of myoglobin, is an excellent oxygen carrier and oxygen storage unit. When it comes to structure, on this slide you can see the structure of heme. So you can see that it is substituted porphyrin ring. We just covered porphyrin ring. So porphyrin ring that has substituents on each ring. And at the center of the porphyrin ring, iron 2 plus ion is coordinated to 4 nitrogen atoms. Now iron 2 plus actually has 6 coordinative sites. So it can coordinate to 6 atoms. One coordinative site is occupied by histidine molecule, by histidine residue, histidine side chain. So histidine from F8 chain, histidine from F chain, that's 8, histidine is 8th amino acid on F chain of myoglobin. So that occupies one coordination site of iron. 4 coordination sites are occupied by nitrogen. And that will be the case in deoxymyoglobin. If oxygen molecule complexes to iron, then we have a molecule of myoglobin, and that occupies 6th coordination site of iron. Histidine, 7th amino acid, histidine from chain E, coordinates with that oxygen and stabilizes bonding of oxygen to iron and stabilizes the entire myoglobin, oxygenated myoglobin molecule. You can see on the right more detailed description, more detailed structures and how stabilization works. So histidine F8 electron pair from nitrogen is donated to iron to satisfy its coordination number of 6, while histidine 7 from chain E through hydrogen bonding stabilizes oxygen molecule that is bonded to iron. You can see that porphyrin ring has 4 pyrrole rings, while the active site of myoglobin has imidazole side chains, has histidine side chains that contain imidazole. Imidazole and pyrrole look alike. They are 5-membered rings with nitrogens, but imidazole has 2 nitrogen atoms, and that makes its properties rather different. It has different acid-basic properties, and that's what makes it suitable for participation, for being in an active site like this. And in fact, you can see how 2 different nitrogens operate. One donates free electron pair, while another one is engaged in hydrogen bonding. Here is structure of myoglobin. Myoglobin means oxygenated myoglobin, one that is bound to oxygen molecule. And I think I took this from Wikipedia. I may have rotated it a little bit to fit the drawings you have seen previously. Two oxygen atoms are those two big red spheres, and you can see flat porphyrin ring. And also you may see two carboxyl groups, those that are on the left side of the drawing. So carboxyl groups with two oxygen atoms. So you can see those carboxyl groups are oriented outside towards the solvent. While at the opposite side of porphyrin ring, there are allele groups. Allele groups are not that readily visible here, but they are easily seen in the previous drawings. And those allele groups, through hydrophobic interactions, interact with interior of myoglobin, with side chains of alpha chains of myoglobin. Now we are going to look at binding of oxygen to myoglobin. So here is an equation. It's a reversible process. So myoglobin reacts with oxygen to give oxymyoglobin, or we simply call it myoglobin, and one without oxygen we call deoxymyoglobin. We can define a dissociation constant for this process, equilibrium constant, so it's called in this case dissociation constant, and it's multiple of concentrations of myoglobin and oxygen divided, or deoxymyoglobin and oxygen divided by concentration of myoglobin. And then in turn we can define the proportion of total myoglobin that is bonded to oxygen, and that's called fractional saturation, and it's labeled as Y. So myoglobin, concentration of myoglobin divided by total amount, deoxymyoglobin plus myoglobin, concentrations of deoxymyoglobin plus myoglobin. Or if we substitute expression for K in this, then it's concentration of oxygen divided by dissociation constant K plus concentration of oxygen. It's important to use constant K because constant K tells us what is the affinity of myoglobin for oxygen. Obviously, large constant would mean that equilibrium is in this case, actually, large constant, the way it is written, would be low affinity, and small constant would be high affinity, because the way it's written is as dissociation constant, as myoglobin dissociating to deoxymyoglobin and oxygen. Now, usually we don't represent amount of oxygen as concentration of oxygen, but rather as partial pressure of oxygen, because oxygen is a gas. So typical expression for fractional saturation is partial pressure of oxygen divided by dissociation constant plus partial pressure of oxygen. And we can plot oxygen myoglobin, oxygen binding to myoglobin. So if we plot fractional saturation and partial pressure of oxygen in torus, we can see that oxygen binding or saturation follows parabolic curve. So with increase in partial pressure of oxygen, there is parabolic increase in saturation of myoglobin. So myoglobin, as you can see, actually does have rather high affinity for oxygen, because even small increase in partial pressure of oxygen will result in considerable increase in saturation of myoglobin. And we can define, and here is a curve for binding of hemoglobin to oxygen, and you will see that curve exhibits S shape, or is S-shaped curve. You will see later why that is the case. And we can also define 50% saturation, partial pressure at which there is 50% saturation of myoglobin, or, as it will be later important, hemoglobin. And that point equals K. dissociation constant K, and sometimes it's called P50. You can see that 50% saturation is much lower for myoglobin. Myoglobin is 50% saturated at only 2.8 torr, while hemoglobin is 50% saturated at 26 torr, much larger value. And that makes sense because hemoglobin has to transfer oxygen to myoglobin so that it could be used inside the cell, so that myoglobin can store it inside the cell. So it makes sense that myoglobin has much higher affinity for oxygen compared to hemoglobin. That allows hemoglobin to transfer it to myoglobin. Note that at 50% saturation of myoglobin, affinity of hemoglobin for oxygen is almost negligible. Now we are going to look a little bit at the structure of hemoglobin. Both hemoglobin and myoglobin are transport proteins. Their role is to transport oxygen. But structure is rather different because hemoglobin is much more complex. It is what we call a heterotetramer. So it has quaternary structure, and its quaternary structure is that of a tetramer. Tetramer means tetra, 4. It's composed of 4 chains. And those 4 chains are 2 pairs of identical chains, 2 alpha chains and 2 beta chains. So we label hemoglobin or indicate structure of hemoglobin as alpha 2, beta 2. And you will see later that that's important for its function. This structure is important for its function, that there are 2 alpha and 2 beta chains. These chains each of these chains resembles that single globular chain of myoglobin. Tertiary structure is very similar. It's not identical, but it's very similar. For example, alpha chain has 7 alpha helices instead of 8 of myoglobin. So alpha chain lacks D helix of myoglobin. But otherwise, if we were to look at it from the outside, structures are very similar. On the other hand, primary structure, sequence of amino acids, is very different, that of myoglobin and hemoglobin, and they are only about 18% identical. Now, when we consider oxygen binding to hemoglobin, it's actually the same situation in hemoglobin and myoglobin. But for hemoglobin, consequence of oxygen binding plays crucial importance for its role and for its function. So, in case of deoxyhemoglobin or deoxymyoglobin, when oxygen is not bound to heme, iron of heme is actually below the plane of the heme. So iron is attached to 4 nitrogens, and it's attached to histidine of the F chain, so histidine F8. And that bond to histidine basically pulls iron in this presentation down below the plane of the porphyrin ring. So as you can see, it's more of tetragonal pyramidal shape or conformation with respect to iron rather than planar, the regular pyramidal shape. Actually, it's kind of distorted tetragonal pyramid rather than regular tetragonal pyramid. Now, when oxygen binds, binding of oxygen causes iron atom to move up into the plane of the porphyrin ring, because now iron has 6 ligands, and it assumes regular octahedral shape. As iron atom moves up into the plane of porphyrin rings, it carries with it side chain of histidine, because it carries out imidazole ring up with it, and that means that chain of histidine moves up. But that's because histidine is part of the F chain of or F helix of hemoglobin or myoglobin. then the entire helix has to move. And it does, entire helix moves. But since this is one large chain, all those helices, all those 7 or 8 helices are all part of the same globular protein. If one of those helices moves, the rest of them have to move to accommodate that movement, which means that binding of oxygen to heme causes considerable conformational change of the entire chain. In case of myoglobin, that is not a big deal, but in case of hemoglobin, it is, because when that chain changes its conformation, it's also attached to neighboring chains. It's part of a tetramer. And so it will cause neighboring chains to change conformation, and that will affect also their reactivity. About a minute ago, we have seen curves that show binding affinity of oxygen for myoglobin and hemoglobin. And you have seen that behavior of myoglobin is relatively simple. Affinity curve shows very simple hyperbolic behavior. On the other hand, behavior of hemoglobin is much more complex compared to that of myoglobin. Not only that hemoglobin has lower affinity for oxygen, but it also exhibits sigmoidal dependence, or an S curve, when it comes to its affinity for oxygen. And the shape of this curve is actually a consequence of what we have just discussed. It's a consequence of conformational change that is a result of binding of oxygen to heme of individual chain. And so we call that cooperative binding, sequential binding model. Actually, those are two different models, and we use combination of those two models to explain affinity of hemoglobin for oxygen, or to explain shape of a curve that represents affinity of hemoglobin for oxygen. So this S curve or sigmoidal curve. So that's cooperative binding and sequential binding models. To explain affinity of hemoglobin for oxygen, we use models that are actually originally developed, to explain activity of enzymes. Hemoglobin and myoglobin are not enzymes. They don't catalyze any reaction. They are transport proteins. Nonetheless, their behavior is similar and could be related to that of enzymes. And model that we use, cooperative binding model, relies on explanation that is consequence of change in conformation. According to that model, any protein that is composed of individual subunits has those subunits that can exist in two forms, in T form or R form. R form stands for relaxed form, and that's the active form. So in this case, R form is one that is capable of binding oxygen. T form is taut form, or it is also called tense form, and that's inactive form. That one has little or no affinity for oxygen. So here they are schematically shown. So deoxyhemoglobin is shown as existing here. So we have a molecule of deoxyhemoglobin schematically shown as existing in T form. So very low affinity for oxygen, and you see it's surrounded by oxygen molecules. You can see this purple, so this is square-shaped T form. This purple one indicated in R would be R form, relaxed form. That's the one that has high affinity for oxygen, and that would lead eventually to hemoglobin or oxyhemoglobin. So at low partial pressure of oxygen, because this is equilibrium reaction, hemoglobin has very low affinity for oxygen. But as partial pressure of oxygen increases, affinity of hemoglobin very slowly increases. Affinity of hemoglobin for oxygen very slowly increases. You have seen that from that curve that there is very slow increase in affinity until it reaches a certain critical point. And at that point, there is very sharp increase, and then affinity plateaus. That's that S curve. So this is what we are going to explain now. So we have already seen there are two forms, T form and R form. And so now deoxyhemoglobin is in T form. Now, due to thermal motion, some of these subunits, there are two alpha and two beta subunits. So some of those subunits may simply at random assume R form briefly, and that R form may bind to oxygen. If we look at these chains, it would be alpha chain that would do that. And in fact, alpha chains, even in T form, have some very low affinity for oxygen. Beta chain have much lower affinity for oxygen. That's why we have two types of chains in hemoglobin, alpha and beta. As I said, as a result of thermal motion, it is possible for a chain to flip from T to R form when it has high affinity, and then that R form would quickly capture oxygen molecule, and that would result in change in shape and binding to oxygen and all of that. Alternatively, oxygen could even bind to T form of an alpha chain. Constant is low, this is a reaction constant or dissociation constant is high, but nonetheless it's equilibrium process, and if a lot of oxygen is present, then this could occur. So what could happen is that an oxygen atom binds to alpha chain, which has relatively higher affinity for oxygen. Well, binding of oxygen will cause that alpha chain to flip into hemoglobin, from deoxyhemoglobin into hemoglobin chain that is in R form. And because it's in contact with neighboring chain, it will cause neighboring chains also to flip into R form. And those chains, of course, don't have any oxygen bound to it. But now they have high affinity for oxygen. And so oxygen is going to be bound to first alpha chain, which will cause remaining beta chain to flip into R form. And now in R form, beta chains also have high affinity for oxygen, and they will also bind to oxygen. And very quickly, hemoglobin will be fully saturated with oxygen. And so you can see now why we have sigmoidal curve. And the same process here is explained step by step. So first we have all 4 subunits of hemoglobin in T form, and then as one flips to R form, that's that slow process, that almost linear, very slow increase in the slope of the curve, where with increase in partial pressure of oxygen, there is very low increase in affinity of hemoglobin for oxygen. But now, when partial pressure of oxygen has reached some critical point, then enough of the alpha chains have bound to oxygen, have formed a bond to oxygen, that there is noticeable number of hemoglobin molecules that have one oxygen atom and bound to them, and then that results in flip of the neighboring 2 chains to R form. And that's where that sharp increase in slope of the curve starts at this point. And so this is very fast process. Flip of the 2 chains, neighboring 2 chains to R form, binding of oxygen to second alpha chain, and that flips remaining beta chain to R form. All that process is very fast, and then binding of 2 oxygen atoms, first one and then second to beta chain, and that's where we're reaching the plateau part of the curve. So cooperative binding and sequential binding. Cooperative binding means the chains cooperate, and sequential binding, that one oxygen molecule binds at a time. So that explains sigmoidal behavior of hemoglobin, of affinity of hemoglobin for oxygen. Hemoglobin has very high affinity for oxygen. That's hemoglobin molecule that is bound to 4 molecules of oxygen. And as such, it wouldn't readily release oxygen to myoglobin. Under physiological conditions, hemoglobin must release its oxygen to myoglobin so that myoglobin can store it inside the cell and it can be used for needs of the cell. As I said, on its own, actually, once fully oxygenated, hemoglobin has very high affinity for oxygen. But there are several phenomena under physiological conditions that will cause hemoglobin to readily release oxygen, and it releases it in reverse process, in basically reverse sigmoidal curve. And first of them is effect of protons. It's called Bohr effect. So first, as a consequence of conformation change when oxygen is bound, protons are released from molecule of deoxyhemoglobin. So when oxygen binds to hemoglobin, protons are released, and that results in more acidic environment. And of course, then, since process is reversible, presence of protons, excess of protons, low pH, will result in reverse process, because those protons will bind back to molecule of hemoglobin, but if they bind back, that will cause hemoglobin to release oxygen. Consequence of it is that at low pH, hemoglobin has lower affinity for oxygen and releases oxygen. That's simply Le Chatelier's principle. And so this reduced affinity of hemoglobin for oxygen at low pH is called Bohr effect. One variation on Bohr effect is effect of carbon dioxide. As a part of respiration, tissues release carbon dioxide. This released carbon dioxide is of course dissolved in aqueous environment, and it reacts with water. Now, reaction with water ordinarily would be very slow, but there is an enzyme, carbonic anhydrase, which considerably speeds up that reaction. So carbon dioxide reacts with water to form carbonic acid. And of course, carbonic acid as an acid is source of protons, and these protons make environment more acidic. Which means that as part of respiration, tissues release carbon dioxide, that will in turn, through release of protons, cause hemoglobin to release oxygen. And we also have an effect of chloride ion. Chloride ion forms salt bridges in molecule of hemoglobin. And those salt bridges stabilize chains of hemoglobin, basically. Now, chloride ion binds better to deoxyhemoglobin than to hemoglobin. In other words, chloride ion stabilizes T form of hemoglobin over R form. So, presence of higher concentration of chloride ions will cause hemoglobin to release oxygen. Now, the three effects that I mentioned already are very important, and they reduce affinity of hemoglobin for oxygen and cause hemoglobin to release oxygen. Those are effect of protons, carbon dioxide, which then boils down to effect of protons, and effect of chloride ion. But they are not nearly enough for hemoglobin to release oxygen so that myoglobin can pick it up. And there is a metabolite that plays a crucial role. That metabolite is 2,3-bisphosphoglycerate, or BPG for short. And that one is arguably the most important factor that regulates oxygen binding activity of hemoglobin. So, BPG ends up binding to ends of beta chains. And when it binds to ends of only beta chains, and this is very important because beta chains are the ones that in T form have absolutely no affinity for oxygen. Alpha chains would still have some affinity for oxygen. So if BPG were to bind and stabilize T form of alpha chains, that actually would not really accomplish the task because alpha chains would still have some low affinity for oxygen and they may not flip from R to T form. But beta chains in T form have absolutely zero affinity for oxygen. And that's what BPG does. It gets in between the two beta chains, binds to them, and fixes them, basically forces them into T form, forces them to release oxygen and to flip into T form. And then basically, as long as it's bound to them, freezes them into that T form. And that, of course, causes neighboring two alpha chains also to flip to T form, and that reduces affinity of hemoglobin for oxygen, and hemoglobin releases all of its oxygen quickly. So this is the most important factor that causes hemoglobin to release oxygen to myoglobin. Now, what is interesting is that fetus, of course, does not have respiration of its own. It relies on mother to perform respiration, and so that to get oxygen. But that oxygen in mother's hemoglobin is actually transferred to fetal hemoglobin to placenta. And fetal hemoglobin actually does not have beta chains. It has gamma chains, which have absolutely no affinity for BPG. They are unaffected by BPG. So 2,3-bisphosphoglycerate will cause mother's hemoglobin to release oxygen, but since fetal hemoglobin is alpha-2, gamma-2 hemoglobin, it has gamma chains instead of beta chains, it will pick up that oxygen and then carry it to its own cells. So that's the reason why fetal hemoglobin is different from adult hemoglobin, because fetus does not perform respiration and has to rely on basically getting oxygen, taking away oxygen from mother's hemoglobin. Okay, and this completes this lecture on structure and function of myoglobin and hemoglobin.